Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists,
Louise Cole talks about how it's fungi that got her started off on her biology journey.
I loved all the mycology, all the fungal biology with John Webster, who was the academic and doing a lot of research.
The tendency of using microscopes less and less as you become more senior.
I'm very fortunate if I get to sit at a microscope once a fortnight.
And what it was like for her when she hit the glass ceiling.
I was at a senior lecturer level, and I was told there wasn't scope for me to go to associate professor or professor in my role as a core facility manager. Even though I was successful in getting philanthropic or external funding.
All in this episode of the Microscopists. Hi, I'm Peter O'Toole from the University of York, and welcome to this episode of the Microscopists with Louise Cole from the University of Technology in Sydney. Good morning, good evening. Hi, Louise, how are you?
Good evening, good morning. I'm good. How are you?
I'm good, thank you. So actually, you're quite a good guest actually to have on this for many reasons, because you've obviously gone through an academic route, moved into the world of facilities. You're going on to do a lot of charity work with Australian microscopy and microanalysis. So there's quite a lot of ground to cover, and I think it will be good for the listeners and viewers to actually hear how that career has changed, evolved. I think there's actually quite a lot of evolution we'll talk about throughout today's talk. But firstly, currently, I don't know the answer to this. How big is your facility?
How big? In terms, we have 10 microscopes, optical microscopes, and we have about 175 users a year and two staff.
So you've got 10 microscopes, all light microscopes or some electron microscopes?
Yes. No, all optical. There is a electron microscope facility, and that's the microstructural analysis unit. And that's really focuses on material science. So not really biological at all.
Okay, because from your background, you're definitely someone who spans both light and electron microscopy. And there's a growing CLEM market. I can think of some very good examples of people who do light and electron microscopy, but there's not many that span those two technologies.
No. Yeah, my background. So I started off as an electron microscopist, and that's how I was trained. And I still have a huge passion for electron microscopy. But in terms of when I started working in a core facility, it was light and laser area. So, and that was at Sydney Uni in what was then called the electron microscope unit. But they at the time, they also had a light and laser unit with a multiphoton, a couple of confocals, an old MRC 600. That was what I was actually using that for my own sort of postdoc research. So I entered the light and laser area, but also I was involved because I had an EM background as well. I also helped set up the cryo-EM pipeline with a high-pressure freezer and the AFS free substitution unit as well. So I was sort of helping the biologists prepare their samples for EM as well.
This is cryo-EM not for structural biology type EM, but cryo-EM for structures of cells and
membranes. Yeah, absolutely. Yeah, because of the artifacts of chemical fixation, it was really it was really focusing on freezing bigger bits of tissue and then looking at the preservation of membranes more so. So, and even though I ended up just managing a core facility with optical microscopes, I actually still love EM and find a way to and either sit on one or we've set up a pipeline for specimen prep for EM, just conventional fix last year here in the facility, because we actually have we also manage an ultra microtome, but nobody really uses it because we don't have that pipeline set up as properly, so that that biologists can use it. So they tend to go to another university or somewhere else, or they have a colleague that does the EM side, because it's quite hard to, you do need staff to set that up. And as I said, the microstructure analysis unit, they're used to material samples, so they don't know what to do with biological specimens. But so I'm working on that. That's still work in progress.
So we've got the EM and the LM, but actually we've just lost our TEM thanks to minor water.
Oh no!
Ingress. Well, it didn't actually ingress. It was just condensated out inside it as it got over chilled. But actually, we're using our nano center, which is more materials focused, because they have some excellent TEMs. So actually, for our use, we can prep and take samples there. We've got for structural biology. It's interesting, the cryo-EM. Now, if you say cryo-EM, most people will jump down the structural biology route rather than the sort of 3D volume or preservation of large structures. As a classic biologist would use cryo-EM. It's hard to, I haven't worked out the best way to word it. I always call structural cryo-EM or cryo-SEM or cryo-TEM. It's not easy.
I know, it's a whole new language. And it's amazing what you can do now with serial block face, SEM. We don't have one here. There's one at Sydney Uni, but in terms of getting your volume EM as well, they're pretty amazing. Yeah.
As is arriving in a month and a half's time.
I didn't know that was.
Yes. So Zeiss Gemini 460.
Oh, fantastic.
Yeah. The team are excited by that one. Anyway,
congratulations.
I presume your first confocal microscope then, was that the MRC 600, or did you use one before
that? So I did use one at Oxford Uni, and that was basically one of the earlier models of Biorad. It wasn't a commercial, they were building them there when Mark Fricker was there. And so my previous supervisor, Chris Halls, promised me a confocal. So I was working with him, doing a master's in plant sciences in South Park Road in Oxford. And I followed him up the hill, literally up the hill, up to Oxford Brookes University when he got a professorship up there. And I followed him up there. And and I ended up doing a PhD with him. And he promised me a confocal microscope when I was, so that's why I came back from overseas travels to do a PhD with him. And the day I submitted my PhD, he willed his confocal in. So never quite forgiven him for that. So, because it took him a while to get funding for the confocal, of course. And he bought a Zeiss confocal. So, so I've never... so the very first one that I spent quite a bit of time on was the MRC 600.
So you actually sent a picture of Chris.
I did. Yeah.
Points out of it as well. So I take it he was visiting you, or were you visiting him at this point?
That's his cottage, just outside Oxford. And that was the time, that was the botanical microscopy meeting. I came back for FOM in London.
Yeah.
So, and went to the botanical microscopy meeting for a bit, and then spent some time with him as well. And he was really retiring at that stage. So still in the same cottage with his partner Kay and their huge Newfoundland dog. So it's always lovely to go. It's always lovely to go and visit him.
And it's actually one of my staff, Claire Steele King, was also a graduate out of Chris's lab.
Absolutely.
And there's a few others out of Chris's lab, and you keep bumping into, Chris. So his was huge.
Absolutely. Absolutely. Yeah, I think it was the good curry parties, and maybe the jam donuts for birthdays. It was a fun place to work, and he was amazing, amazing supervisor who was more like a brother really than yeah. So yeah, so I still I'm still very much inspired by him. He used to come once a week into the lab, and he would put his hands on my shoulders and say, what's new. And so I remember that quite vividly. So I would want to get some data to show him from one week to the next. Yeah, because he was so lovely. But if he ever tried to do an experiment himself, everyone just sort of ran a mile because he was crazy in the lab. When you haven't done experiments for years, I'd be the same now, and you'd go back and you'd want to do some fixation or something like that. So then it would be wouldn't want him contaminating any of our buffers or anything like that. So that was quite funny.
And Chris was obviously a big influence at the Royal Microscopical Society as well. And I don't know if did you have much involvement with the RMS back then when you were in the UK?
So we did a lot actually, because he would often go down the hill to the office, the RMS office, and I'd tag along. So I mean, it was like, this might sound a bit odd, but like one of Chris's, girls, we were very happy to go along and help. So and because we hosted the botanical microscopy meeting as well. I think that was the 5th one, mid-90s. We were all involved in organizing that conference. I had to design a little badge for the RMS, and also the t-shirt for the 5th botanical microscopy. I think I sent you a photo of that. I was modeling. Yeah,
that's what it is. I had no idea what the picture was of. It just said RMS poster girl. I thought, I have no idea.
I know. So I designed that for the RMS, and it's a picture of Robert Hooke's nettle. It's a nettle. I think it's the leaf trichomes of a nettle leaf. And he got me to design the t-shirt and model the t-shirt. And some people still have their t-shirt from that was the mid-90s.
Have you still got yours?
No, unfortunately.
Yeah, got rid of it and now you regret getting rid of it?
I do regret getting rid of it. I don't know where. I mean, it might be at my parents' house. I don't know, because I don't normally throw things out. But that was how involved we were with the RMS at the time. We were more than happy to help and be involved. And we used to go to the meetings in London all the time. I don't know if you still have the one-day meetings in London anymore.
No. And the RMS events have evolved quite a lot as the environment and everything else.
But they're much bigger. Yes.
So there's different meetings. They're generally 2-day meetings, but not all exclusively. There are some ad hoc meetings as well. But the frontiers of bioimaging, I guess, now has replaced the light microscopy life science meetings. So you've got the frontiers of bioimaging that come forward. And then you've got the big MMCs, Microscience, now Microscience Microscopy Congress, which is now so much bigger.
Yeah, that's a great conference. Yeah,
encompassing for all the different science disciplines, life science, all the different life sciences, including the plant, microbiology, through to the material side.
Yeah, it's amazing, isn't it? Because they were one-day meeting all those many moons ago. And Chris would get a bit annoyed because we'd miss the very last couple of lectures because we'd go off to the National Gallery to try and get some artistic influence, and we'd be back, but we would just have a little side visit, feeling more inspired and energized from a little diversion.
That's why you always went disappearing. So you said that you never throw anything away. Does that mean you're a hoarder?
No, I don't. I was going to say I don't tend to certainly don't throw any microscopes away or microscope parts away. So you never know when you might need them for something. But maybe I'm more selective at what I purchase, and hopefully I look after it well so that it lasts the test of time.
So we've got a big cupboard with lots of parts of microscopes.
I do. I have still bits of microscopes on the floor around. Everybody seems to give you them though, don't they? Because they just assume that you need them. And I do say no now to some microscopes, that some little benchtop ones, because we don't have the need for those. We are in like an advanced microscopy facility, so we can't. Yeah. So, but you never know. What about you?
Not so big. We used to keep bits, but I think we've started to run out of space as the lab grew and grew and grew. And so then we had to get harder. And we realized that actually for the user base, they don't need sort of temporary fixes. We need to make sure everything's properly serviced, everything else. So generally, we're pretty good at moving things on. We do keep some parts, obviously, but we donated our LSM 780 to Yobe State University, so Mahmoud Maina and by RTC over there, with all the spare parts of another system. So we'd shredded one system in case ours broke because it wasn't supported fully anymore. Then we got a replacement, so we donated that with the spare parts, which is just as well because the laser's just on the blink and they have a whole spare laser. But in defense of Zeiss, we also asked Zeiss, and they said, oh, we're removing one at the moment. We'll send you that laser as well. And then,
fantastic.
Then Mahmoud is actually couriering that. So they've got another laser now. So it's really good. But the other systems we quite often, actually through Claire Steele King, did a lot of the initiation or drove forward. We sell quite a lot of stuff onto Unigreen.
Okay.
Sort of they take the equipment and then sell it on, so it's like a second-hand or swap shop. And actually, oh gosh, Stuart at work, a new academic, has just bought 2 stunning microscopes for hideously low prices. Really good top dollar microscopes. So I'm now encouraging everyone to sell more to Unigreen and to buy from Unigreen to make sure it's completely cyclic. And so actually we don't need to keep the parts because we know we can go to Unigreen to get spare parts should we run out of them.
Fantastic. I think I need to look into that.
That's certainly something to, do have a consider. So your facility, so we talked about the facility, the first light microscope. Take you back even further. When you were a child, what was the first job that you can remember wanting to do?
Ooh, I think I know I wanted to travel. So I did. I might be ashamed to admit this. I did want to be an air hostess because that was my gateway to seeing the world. So wasn't thinking about learning different languages. I was just thinking about how can I get on that plane. So I know that was one of the first jobs that I wanted to do. I don't think I took it any seriously sort of after that, but that was I just think I just wanted to see the world, and actually I still want to see the world.
Were you British to start with?
Yes.
Yes. Okay.
Yes, I am. I'm still British.
Yes.
I'm a permanent resident here, but you might hear an Aussie accent. So
definitely an Aussie twang in there, that's for sure.
But I think that's the Bristolian in me. I think that's because I'm from the southwest of England, and Bristolians have that intonation that they go up at the end of their sentences.
Okay.
So which is what Australians do. But so I go back to, obviously I went back to England recently, and everybody there, most of them said that no, they couldn't hear the Australian, but then other people can. And then when I speak to Australians here, they just say I'm very English, so I don't mind. I used to say that that when my eldest when he lost his British accent, well, because he started, even though he was born in Australia, because he was obviously imitating our accent, and my husband's English as well. So he sounded quite English. And so I thought as soon as he starts sounding Australian, we were going to pack our bags and leave. But we're still here 27 years later. So
So they are proper Aussie-born, Aussie-bred.
Yes, but they also have British passports, so they're very, very happy to be British as well. So it gives them that freedom.
And you do send some pictures, so I presume this is the slightly like, and their partners, I presume.
Absolutely. So my husband is there on your right. That was his 60th birthday last October. And I'm obviously the one with the red shirt. So the Finnegan is in the middle. So he's my eldest, and that's his partner Naomi. And then so Rowan and it's my second son, and Angelina is his partner as well. So actually we're going for dinner. It's the family dinner. It's the family dinner season now, because it's we have birthdays in August, September, October and November. So we all go out for a family dinner, and it starts tomorrow night.
It starts tomorrow night, did you say?
Starts tomorrow night, yeah.
Okay. What sort of food are you going for?
So tomorrow we're going for Australian with a French twist.
Okay. I'll take your word for that.
Yeah.
I'm trying to think what that could be. Snails on barbecue? I don't know.
I think it's so it's more French with an Australian twist, I think. I'm from the menu. I haven't been there before. So it is a couple of up and coming French chefs who have got quite a big growing reputation. And they've taken over a pub, and they've turned it from a gastro pub into a nice restaurant. So and when it's your birthday, it's nice to go for something a bit more fine dining. So they have focused more on using Australian bush foods. So lemon myrtle, and so different, so more Australian herbs and spices to a French dish, I think. But I won't know until tomorrow.
You cook as well, I believe, because you also sent a load of photos of food.
I did. I'm such a yeah. Absolutely. If you look through my phone, it's either the pictures of microscopes or food that I've baked. So I am a keen baker, but that's because I don't do experiments anymore. I love to follow. I actually follow recipes, and I actually have over 100 cookbooks, and I have culled them recently. That was. I know I have culled them. I'm quite easy to buy for as a present, so long as you don't buy the same cookbook that I've already got. So I do love having people over for dinner. So that's where I relax, really. Saturday I could spend all day cooking. The only thing is everyone's on diet, so I'm not doing it less and less now. And the bigger the showstopper dish, the better.
So what was your signature dish?
So I think, well, there's the ragu right behind you. Yeah. And in the bottom left-hand corner, that's easy. That's I always do a ragu, a butter chicken. For the veggies, I do there's the falafel, eggplant parmigiana. And at the moment, it's because it's winter, it's just a veggie tray bake, with about 10 veg is the easiest thing that I cook. So so I do have a bit of a reputation. All my friends do like, to come round. We're not really allowed to go out for dinner. I have to cook, but time, obviously sometimes I just don't have the time, because I do have to take the day off before to prepare, unless it's at the weekend. It's and also I have to prepare the table as well. 2 tablecloths, crystal, very grand. So I'm either 100% in or not at all.
Okay. And you also do cakes, I presume?
No, that's actually one of what I would call a showstopper. That's a Iranian dish called a tashin. I don't know. Sorry if I pronounced that.
Yeah,
tagine. It's a tashin, which means, I think, TACHIN. So it's inverted. So it's rice which is baked with saffron, and it's got egg and yogurt which lines the dish, and then you fill the well. So you make like a trough, and then you put in the mincemeat, and that's cranberries, So it's like a Persian dish, and then you cook it and then you flip it out. So yeah, so and it freezes really well. So I had to freeze half of that because I doubled the recipe and it was huge.
Did people just not like it, and that's why you had so much? I'm joking.
They would tell me. They would tell me.
I'm joking. So as we go back to as a child, when you wanted to be an air stewardess.
I know.
So what got you into science?
So at school, I was very good at maths, and I loved biology, but I wasn't sure what I really didn't know what I wanted to do, or study at A level. And I remember a parents' evening, and I was just copying my friend who wanted to do business studies, because I really didn't know what I wanted to do. And I think so that was really a pivotal time, because the biology teacher said to my parents, she's good at biology, she should do biology. And I went, all right then. So yeah, the rest is history. But I did biology and maths and chemistry at A level. And I loved biology, but I didn't like dissecting the rat at all. So I loved all the plants. So I ended up studying botany at Exeter University, carrying on and working in plants. So I stayed in plants for quite a while.
And then over to Oxford for your PhD at that point, or master's or PhD?
So I did masters first of all, because when I finished uni, I loved all the mycology, all the fungal biology, with John Webster, who was the academic and doing a lot of research. He wrote a book on fungal biology, and that's where I really became interested in microscopy, because obviously there were microscopes in the course that we could use for the practicals. And I was fascinated, and I would always. So I met my husband at university. He was studying mathematics, and I always would spend the night before the practical reading up on everything, so that I could spend as much time enjoying the microscopy. So he didn't like the fact that I prepared for the practical the day before. So I was offered a PhD by John Webster when I finished my undergraduate, studying aquatic hyphomycetes, which are the fungi that live on the foam of rivers. Mm-hmm. So, which of course we all know about, and we know how, they are important, but I decided that I needed a bit of a break. So I turned down that PhD and then took some time off, and then applied for a research assistant position working with Chris Hawes on an endocytosis in plants at Oxford Uni. So that's where my endocytosis and microscopy journey really began. So Chris was a Zeiss snob, really, in terms of, and I think that, that's a very fond way of saying that he loves Zeiss microscopes, and he would only buy sort of a Zeiss microscope. So my first Zeiss first microscope that I for my master's was an Axiophote or Axiopho. How do you as I Yes, I say pot too. So that was I used that for my fluorescence microscopy of looking at the uptake of lucifer yellow or fluorescent dextrans as markers for endocytosis in plants. So, and as well as the EM, but I did more EM than light at the time.
So what was your first EM?
So that was a Joel. I can't remember the number, but that was a Joel. Yeah. So a Joel. So and that was a 10. Yeah. So.
Okay. Yeah. I'm going to say, because this may have been a Zeiss snob, but actually, like you said, we've got a lot of Zeiss products, but our TEM was a Thermo, our SEM was a Joel, and we've had other Joel systems as well. So actually, we do deviate. It's very much.
I'm going to I think exactly. Oh, I think Chris was very he was very happy to admit that. Anyway, so everyone has their favorites as well, and I think he's always had a good relationship with the company too. So, but I mean, people have favorite cars.
Well, yeah, actually, we do have the same brand quite often. I wouldn't say I'm the biggest, but we like the cars. They're just good value. But there you are.
Absolutely.
I had all the money in the world. Might not be what I had. I might change from what I've got on the driveway.
True.
So that got you into the microscopy side, and you were through. So why? I guess because your love of travel, but is that what enticed you to Sydney?
So, so I attended a conference in Vancouver, and here was my.
Louise's lights in her office have just popped off because of an automatic timer, and she's obviously not moving around enough.
No, of course. So, yes. So, yeah, so there was a conference in Vancouver, Canada, that I wanted to go to, and that was the International Mycological Congress in British Columbia. And again, so going back to Chris, I remember that he would not, as a supervisor, didn't have the money. Well, he said he didn't have the money to pay for me to go. So I said, okay, that's all right, I'm going to go anyway, because I really, really wanted to go. And I hadn't been to that part of the world, this beautiful part of the world, Vancouver. So, but I called his bluff, because he said, okay, I'll pay for you then. He didn't want me to go with my own money. So I met Anne Ashford, and Bill, her partner, Bill Alloway, and they were working on the pleomorphic vacuoles in a eucalypt mycorrhizal fungus. And they were so they have these amazing tubular vacuoles which were responsible for long distance transport. And at the time when I was looking at endocytosis, because we in plants, I was using a drug called probenecid, which is used to treat gout. And that perturbs the anionic transport system. So, and what I found at the time with plant cells, that a lot of the probes, the fluorescent dextrans, they had free fluorescein. So you would get premature uptake of the fluorophore instead of the fluorescent dextran. So it wasn't a great marker for endocytosis, because the dextran part is 70K. So I was the first person to publish this data on probenecid and how it affects the anion transport system. And she was interested in applying a similar approach in fungal hyphae. So I hadn't quite finished my PhD. I had another year. Had another year or so to go. But she wanted me to do the same experiments on the fungal hyphae. And so she offered me a postdoc and waited for me to finish. So that has actually happened quite a bit in my career trajectory, that I have happened to be in the right place, or where I've been offered a position. So I didn't map it out. I've just been really fortunate to be connected, or it's just, it's more of a timing and having that communication and thinking I'm ready to go, and that's sort of what's happened. Or I found a really good lab and I've waited for them to get money and say, I've told them I'm interested and let me know. And so I came out to Australia in 96 with my husband was offered a job in Tokyo, and I was offered a job in Sydney. And I said to him, well, I'm going to Sydney. We'd been to Sydney for 4 days on our honeymoon. We'd had sort of done a round the world trip. So we knew we liked Sydney. So I said, and it, in hindsight, I think my options for work in Tokyo probably then would have been quite different. So so my husband was fine with it. So I had submitted my PhD, and within a month we'd moved to Sydney, Australia. And the idea was, because it was a 3-year postdoc position, so the idea, so we thought we'd stay for 3 years, stay for the Olympics in 2000, then go back to the UK. So I'm now 28 years into my 3-year plan.
Will you ever come back to the UK, or do you think that's it now? You're staying?
I think we've always planned to come back at some stage. We know that. So at the moment, my youngest is he's going to turn 21 this year. He's got 2 more years of uni. So the reason to go back is to be closer to family. So but I'm not sure I could cope with the weather. It's rather nice out here.
Will your boys come back to the UK?
I think they would like to work in the UK or live in the UK for short periods of time. They do love Australia. So they do talk about going to the UK, whether they will just go back to the UK and stay. I'm not so sure. Again, I think they like the weather here.
That's tough for you, because you're coming back to be close to family, but then if they move back to Australia.
I know, but I think we're more likely to go back before they do.
Okay. So you may come back for a short period and then hop back to Australia.
Yes, exactly, depending on what they want to do. But at the moment, so we have a few more years. So, but I picked up this saying from a friend of mine who her parents said to her, 24 and out the door, which is, that's when you have to leave home, when you're 24. So I said that a few years ago to my eldest, and he moved out with his girlfriend when he was 24. So my 20-year-old son knows he's got a few more years yet, and he's got like a basement, he's got like a man cave, like almost a granny flat. So he's very comfortable, and the cost of living crisis here in Sydney is crazy.
Yeah, okay. It'll be interesting to see how that pans out in a few years' time. So thinking of that, so you've got into working at postdocing in Australia. So you've done the PhD, you've done a postdoc career, but then you moved into facilities at that point?
Yes, yeah. I loved being a postdoc. I honestly thought I was going to be an eternal postdoc. I also thought that I did want my own lab, my own research lab at one stage. But it was very difficult to get funding in that area of plant cell biology. I didn't really have my own research focus. I did try a couple of times to try and get some fellowships. So when my second child was born, that's when I decided I needed to do, because we don't have any family support here in Australia. So I decided to do a more 9-to-5 job, which wasn't, of course, it wasn't a 9-to-5 job. So that's when I took over as light and laser optics manager at the electron microscope unit in the University of Sydney. So that's when I went into managing that part, the light and optical. So I was there for 2 years, but I was involved in 100 research projects across and so it was very, very busy. So and then I was offered a position at what was called the Bosch Institute at the time, in the School of Anatomy and Histology at the University of Sydney, to run their advanced microscopy facility in the Bosch Institute. Now, the Bosch Institute was a virtual institute. So it was the research arm of the School of Medical Sciences. So a colleague of mine at who managed the micro CT at the EMU, basically said, I think this position would be great for you. So I was invited to apply, and then I moved there, and I was there for 11 years, and I really built it from the ground up. And that's where I still remember where I was when I realized I'm not going to have my own research group. I realized that I enjoyed learning about other people's projects, other people's scientific area that they're researching, and I still learn every day from being able to being involved in those different projects, and training the next generation of microscopists.
How hands-on are you now?
Not very hands-on. So although, because there's only 2 of us, we do have the duty microscopist scheme. So there are 2 days a week when I'm on duty where they are allowed to ring me, and I will go in and sit with them at the microscope. So we do have that. I'm very fortunate if I get to sit at a microscope once a fortnight. But and when I left the Bosch Institute, I actually said to the COO at the time, I said, I need a T-shirt to say a good day is a day at the microscope. So because that's where I'm happiest. But I don't get much time at the microscope anymore with everything else.
You learn
that? Yes, I do. I really enjoy it. So because there's only 2 of us managing the facility, if Amy Bottomley, she's the imaging scientist that supports. So I'm the facility manager and the director. So I have 2 jobs. And so if she's away, then I get to do her job as well. So then I have 3 jobs. And that's when I get to sit at the microscope with a user. So I will do training if I can.
You basically say you've got 2 jobs, 3rd job, but we haven't touched on, you are now president, fellow president of the Australian Microscopy and Microanalysis Society, which you sent another picture of here.
Yes, with Nesta.
I don't know what the award is that's on my shoulder.
That's the David Cocaine Award, who was the previous director of the EMU at Sydney Uni. So yes, so we have a number of awards within the society for either for research or service, or special awards for contributions to the advancement of microscopy. So so Nesta was awarded that. So in Perth at our last big national meeting. So
how do you find the time to do that and your other 3 jobs?
So usually weekends is when I put my AMS hat on and do those duties. So, but I really do enjoy it. So the Australian Microscopy and Microanalysis Society it's a wonderful community. So I was president of the Light Microscopy Australia before I became the AMS president, and I wanted to do it because I really was missing the EM side as well. So because the Light Microscopy Australia is a special interest group under the parent organisation of the Australian Microscopy and Microanalysis Society. We have other special interest groups as well. So it's a wonderful community. So I do have to, you have to obviously chair all the meetings as you know, but it's it's where you can really work at the coalface and try and make a difference and inspire others. So I think I'll be doing that for another couple of years anyway.
I do notice over your one shoulder what looks like an RMS certificate.
It is. So yeah, so I have a certificate of fellowship because I was involved with the RMS for many years ago. So so well spotted. I did blur it. I did wonder whether you'd still.
Well, the RMS logo is good for me. Even my eyesight, you can see that a mile off. A bit of
Yeah, so
it'll be crystal clear.
yeah. So when I was with Chris, yeah, he recommended that I apply for that. So yeah, so I've got it very proudly there. And actually, I've just added my initials to my LinkedIn page as well. So we should do something similar for AMS as well.
Yeah, and it's quite good. So actually, I know we've been talking outside of this about some of the joint collaborations for the RMS, because the RMS is there to support internationally as well, and some of the things we can do in common. So instead of duplicating efforts, even if it's copying what you're doing, but instead of copying, taking the materials, rolling them out, and we're talking to Laby as well around those, so Latin America bioimaging. So I think it's nice. I think we're all getting more collaborative now. I think there was a time where actually people became more insular, and I think it's now becoming far more collegiate throughout the different communities, and long may that continue and develop.
Absolutely. Here.
and grow through. Throughout your career, when would you say was the best time in your career? If you could relive a moment of your career, what moment would it be?
I think when I was doing my PhD, the last year or so, when all the data, when all the results are sort of coming together, and then Claire was part of that as well. So Claire will remember exactly what that was like. So as part of a very big group, but a very big family. And I had done enough research in that area that I was really quite across the just the research progress and the knowledge in that field. So I don't know. I mean, I actually felt quite invincible, so at the time, because things seemed to. I remember for my very last chapter, I don't know if you ever met Beatrice Satej-Metra. She's a Golgi person who so she managed a core facility in Gif Servet. Sorry, I apologize to any French speaking person for my pronunciation, just outside Paris. So she managed a core facility there for many, many years after. So she was working with Chris Halls at the time. So and her partner had a wine business. So she would come with champagne on a Friday as well. So we knew how to work hard, but we knew how to enjoy ourselves at the same time. So that was really good fun. And I think that's when I thought that I wanted just to be a postdoc for the rest of my life, because that was fun, because you didn't have a thesis to write after that as well. So I mean, obviously I did with my PhD, but yeah.
They were the best times. On the
Yes.
flip side, what would you say was the most challenging or most difficult time of your careers today?
So I think that's when I'd actually had hit that glass ceiling. And so I was working at so I sort of come to the end of my time at the Bosch Institute. So, and this sort of relates to recognition of core facility staff globally, because so in that role, I was an academic. So, and I don't know how the levels compare in Australia to Europe, but I was at a senior lecturer level. And I was told there wasn't scope for me to go to associate professor or professor in my role as a core facility manager. Even though I was successful in getting philanthropic or external funding, internal funding, publishing papers. So I think I was very unhappy for a couple of years in that role, because I was told that I could, and that still happens here. I'm not sure what it's like in Europe. So the only way to get some career progression, and I still hear it today, because that was for me to change and move universities, and then I went into associate professor role.
So it's not really necessarily an Australian problem. It was a institute problem, probably more than an Australian problem.
Yes, yeah, and I think it was the recognition of even though it's understanding the roles of core facility staff, and I had an academic position though. I could sort of so because we have the academic level, and then we have the technical staff. So there's two streams here. So, and they're very different the way that you go for promotion.
And I'd say the UK is pushing ahead, progressing quite well, but it's still patchy. It's far from perfect. I'm not sure any system's ever perfect for anyone, but there are some good examples. You're very fortunate, always have been very fortunate, but other universities like Liverpool, Warwick, Nottingham are really pushing ahead. Newcastle, actually a very good highlight, are really pushing ahead in these areas as well in the UK, as well as others. But yes, it's still patchy, I would say, at that point. So outside of work, so you've got that ceiling, you have difficult periods. You mentioned before we started, you've got a microscope that's down at the moment and the stress. What do you do outside of work to de-stress? I know this sounds like you sent me some pictures, but go on.
I did. So I have to go to the gym in the morning, just like you do.
Yeah, I think you called it gym junkie, I think you called it.
I am. I have, yeah, but I didn't really. I was never really in a gym before I came to UTS. So when I moved from Sydney Uni to UTS, I was really excited because so again, this was Cynthia Whitchurch. So I joined the gym with her, which was F45. I don't know if you have those in the.
I don't know.
Oh, anyway, you don't need to know. And I joined a coffee club and a WhatsApp group. So it was something that I didn't have in my role at Sydney Uni. I think I cried the very first day I went to the gym. I think I was trying to bench press 3 kilos. And they got me to jump on a box, and I only about a foot high, but I was too afraid to jump on a box. But it didn't take long. So now I go every morning, and it really is I'm the first out of the house in the morning. And it just really helps me sort of center myself. So it might be a strength class, a cardio, or yoga or Pilates. So I can't live without it now. So yeah, and that's Nancy, who was my personal trainer. She always gives me heavier weights every time she sees me, which I'm not really happy about.
You also said, which I had to put on,
yes,
position you call the microscope, because you've just inverted yourself to look like an upright microscope.
Exactly.
You inverted yourself to look like an upright, but there you are.
Yes, the inverted microscopist. So I am actually, so that's on my balcony at home, and I'm training to do some handstands. So getting used to being inverted.
Mm-hmm.
So I haven't quite been able to achieve it without the training support at the moment. But yes. To be upside down for a while is it's very very good for the blood flow.
So actually, if anyone's listening at this moment, we are, I don't know how many minutes into it, 50 minutes in roughly. It's worth just skipping to that bit to see the picture of the inverted microscopist looking like an upright. But I like the fact you also sent me the other picture, and this wasn't the title of it, but you've also done the immersion lens, which was
water immersion, yes, and that's in the same gym. I mean, I will try anything once. So that's 2 minutes ice bath. So which was it's great. It is, but I did 30 minutes breath work before I went in there, which so you're already really, really calm, and you know how to breathe deeply. So I recommend it to anybody to try at least at least once. Well, it's cold enough there. You just have to swim in the ocean, don't you?
I don't do that. I've just come back off holiday where the sea temperature is 31. It's just about okay. My swimming pool is 28. Any colder, I can play.
No, it was invigorating. Yes, highly recommend it.
And you also run, I presume. This is your
Yes, yeah. So we're very lucky in Sydney because it's yeah, amazing. That's the Harbour Bridge, and that's just behind the Opera House as well. So I think anyone would run. So that's on a Saturday morning. If you forget you're running, actually, you're down there, you forget you're running 10K, and let your legs hurt because there's also a seal, a resident seal, just over the other side of that fence that you get to see a seal basking on the steps. So yeah, highly recommend it.
I can't remember we stayed in Sydney, a bit south of the city, and it was very, very friendly running, actually. I found everyone said hello. And that's not always the case. York's pretty good when you go running. Most people will say hello, good morning. When I ran in Sydney, everyone was really friendly. That's rarely the case in most cities. I thought that was...
I think that applies to just the Australian way as well. And I really found that when I first arrived, it's quite strange when you're in the supermarket, just buying your supper, and they say, "Well, how's your day?" And they really want to know how your day is. Mm-hmm. And there'll be a queue of people behind you, but they really want to know how your day is, which wasn't it wasn't the sort of thing that you would respond to in England necessarily. You think, why are they asking me about my day? I'm not going to tell you about. They're very friendly.
Yeah, in the UK, people would say, all good, how are you? And they don't want an answer.
No, they do though in Australia. And it's rude if you don't.
Right, I'll get some quick fire questions.
Oh, goodness me.
First one, are you an early bird or night owl?
Well, early bird now. Although I do go to bed quite late as well, so I don't get a huge amount of sleep. Mm-hmm. So I go both ways, but I have to get up early if I want to go to the gym.
PC or Mac?
Mac.
McDonald's or Burger King?
Oh, neither of those.
What's your favorite takeaway?
We don't have Burger King here. We have Hungry Jack's.
Okay. McDonald's or Hungry Jack's?
I'll say Hungry Jack's.
Okay. Australia or UK?
Oh no. Oh, that's really hard. That's a hard one. I have to say UK.
Okay, if all the Australians listening to this is probably our third demographic. Tea or coffee?
Coffee. 100%.
Beer or wine?
Oh, tequila. Okay. Margarita's my middle name, so has to be tequila.
Chocolate or cheese?
Cheese.
Okay. Electron microscope or light microscope?
Oh, goodness me. I'm going to say electron.
Oh,
I know.
Thank you, considering what your job is. Oh my goodness! So you're working in Australia with light microscopes, and you've just said you prefer the UK and electron microscopes. You've just lost your residency. Anyway,
I hope not.
Eat in or eat out?
Oh, eat in.
And I know the answer to this. Cook or clean?
Oh, yes, definitely cook.
What's your favorite food? No, so we know your signature dish, your ragout, but
yes.
What is your favorite food to eat?
So I'm vegetarian. So I cook meat for my family. So any vegetable. I'm just trying to think of one I don't like. So anything with vegetables.
What about your least favorite food?
My least favorite would have to be boiled egg.
Okay. Hot or cold?
Cold.
Yeah. Okay. Yeah.
You can agree with that.
Cold boiled eggs, but there you are. TV or book?
TV.
You watching any trash TV at the moment? Or what is your
I do watch So so when my husband's out, I will watch. I love all of the craft shows. So Great Pottery Showdown, Great British Sewing Bee. So trashy would be, is it cake? Do you know? Very trashy, but you know, they're very clever. The people that make these cakes, they look like an ironing board, but it's cake.
No, it's a good answer. I wish you sent another picture. I just couldn't get it to work, which is a Barbie cake, but I couldn't get it to.
Oh yeah.
Oh,
sorry. Yeah.
I don't know why.
I organized a microscopy bake-off. And that was a friend of mine, Katie Dixon, who made a cake of me as a core facility, manager. So that was a Barbie in a cake. So there was also a brain and a microscope slide, and somebody else did a tray out of wafers and did some sugar work for the slides.
Yeah.
Pretty impressive.
It would have been quite good next to the brain if you had the brain slices for the histology sections. Anyway, what's your favourite film?
Favorite film? Sorry, I have to do this again.
Lights going out.
Spirited Away, which is Miyazaki.
Favorite Christmas film?
It's a Wonderful Life, of course. I watch it every year. Yeah, beautiful movie.
And what type of music? This will be interesting as a gym bunny. What's your favorite music?
So I like female vocalists. And across all different types of genres. So from country to pop.
Mm-hmm. What do you work out to? Do you work out to that as well?
Whatever they play, which can be pretty horrible. So I know. I don't really know the modern stuff. So something with a fast tempo, I suppose, but you don't go to the gym to listen to good music.
Favorite color?
Yellow.
See, I know you'd seen some of the podcasts, but you didn't realize that if you're saying yellow, you've got to say something like aquadine orange or.
Oh, I don't know.
Oh, no, I know, but no one ever says it. No,
oh, yes, of course. Lucifer yellow. Lucifer.
There you go. Favorite technique?
Oh, I'm going to say light sheet. We don't have a light. I did have a light sheet. I had an ultra microscope. So we were the first in Australia to get an ultra microscope when I was working at the Bosch Institute. Mm-hmm. And I was at FOM in Maastricht in 2013 when Eric Betzig described the light sheet as the rising star. So Stefan Hell was there with his super res, so I was there when that technique really became popular. And I want a lattice light sheet. Have you got one?
We don't have
anyone listening?
The lattice light.
I'm very happy to take it off your hands. Yes.
So we are actually just over the hour, but I have to ask one last question. Do you have any regrets?
Oh, goodness. I think my main regret is that Australia is so far from the UK. I just want to make that. And that would be great, not only for microscopy, but, to be closer to family as well. So, yeah.
That's a good answer. I did have other things I wanted to talk about, but we are up to the hour. So, Louise, thank you so much for joining us today. Good luck in your job.
Thank you.
Moving back to the UK and then moving back to Australia again. Good luck with the presidency of AMN.
Thank you so much.
You're doing there. And everyone who's watched, listened, actually, there's some really good bits through there, and listen to the previous cast to actually cover some of the material that Louise has been talking about in other details. Louise, thank you so much for joining.
Thank you so much. I really enjoyed it. Yeah, enjoy your day.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.